Hello everybody, my name is Aiden Luby and I'd like to welcome you to my presentation on the green synthesis of maleimide derivatives, a potential new acetylcholineesterase inhibitor. Without further ado, we'll hop right into this. So the first thing that we need to talk about here is a little bit of background is what is green chemistry and why you should care. So green chemistry is a metric for scientists to use to critically think about how to do chemistry and chemical engineering in a sustainable and environmentally friendly way. So the table on the left is the 12 principles of green chemistry, which kind of highlights the principles that scientists like to follow in order to be environmentally friendly and sustainable. And then the table on the right shows some equations that we can provide numerical values to our reaction synthesis so we can see how green our reactions are. We will return to the table on the left in just a few slides. But moving on, what is a maleimide, right? So they are building blocks for pharmaceutical drugs and herbicides. The picture on the left is a picture of a maleimide, where you can tell you can put different functional groups on that benzene ring, and their derivatives are on clinical trials for potential acetylcholineesterase inhibitor, which is a Alzheimer's treatment. So you can see the picture on the right is a picture of the acetylcholineesterase inhibitor, which is on trial for Alzheimer's treatment. So here's the synthesis here. I was particularly interested in looking at chlorine, methyl, and nitro functional groups on the ring during the synthesis, and that was carried through 3 steps. The first step was stirring at room temperature, and the second and third steps were utilizing a microwave. And I will show you differences within each step in order to get optimal reaction conditions for the yield. But how exactly green is our synthesis, right? Like, what are we doing that makes it environmentally friendly? Well, steps 1 and 3 have high atom economy, so meaning that the atoms in our starting material are found in our final products. Steps 2 and 3 utilized a microwave heating rather than the traditional thermal heating, which is a picture on the right, which is more energy efficient. Waste reduction, so we only made enough of our products to characterize them and study them. We didn't make excess amount. And then finally, reduction of hazardous reagents and solvents. So we chose reagents and solvents that were less hazardous for us to use and for the environment. Step 1 results. So a couple of notes here is trials 1 to 3 of the methyl group actually used too much starting material. So there was starting material in our final product, which made it less pure. The nitro group is a tough group to work with because it's electron withdrawing, which I'll get into in a little bit. But trials 4 through 6, I stirred overnight. And then trials 7 through 9, I stirred for 45 minutes and left in the freezer. And those are the results right there. At the time of this recording, trials 4 through 9 of the methyl group, I am working on the results right now. So I will have those updated in a bit. But in addition to that, trials 4 through 6 of the nitro group provided the most pure product from the NMR, so they were used to carry out the rest of the synthesis. Step 3 results, again, methyl groups I'm still working on at the time of this recording, but the nitro groups, I changed the reaction conditions of the final step to try and get the optimal reaction conditions for the most pure yield of the final product. And these are the results right here, as you can take a look. Error analysis. So a picture on the big picture right there is a picture of the NMR spectra, which helps us characterize our products. And you can see that we got the endo and the exo product, which was a surprising result. But from a pharmaceutical standpoint, this is not good. We want to be able to control which isomer we get, and the isomers you can see are on the picture on the right here. We also noticed that in most of our experiments, we found some starting material in the final product after analyzing with NMR. And as I mentioned before, the nitro functional group has resonance and inductive effects on the compound, making it difficult to work with during synthesis. So some conclusions. So the identities of the respective products were found using proton and carbon NMR. One of the unexpected results was both isomers in the final product, as I mentioned. Ideally, we want to be able to control which isomer that we get. But the percent yields demonstrate the experiment was successful in general. Future directions. So first, and this goes in priority, the first one is finding the optimal reaction conditions for each functional group in this experiment. So the chlorine, the methyl, and the nitro. The second is establishing control over the isomer, so we're getting the exo or the endo. Improving percent yields and greenness is always an option, and then repeating the experiments with different functional groups. So we want to use Minkowski groups, like anything else really. And then finally, testing the products for pharmaceutical and herbicidal benefits. So that's really far down the line, but we really want to see what are the biological properties of these drugs. Here are the references. I'd like to thank you for joining me on my presentation, and I'll now take any questions you may have.